Many organic molecules are very large and have unique characteristics that allow them to carry out very complex functions in the body. Important categories of organic compounds include carbohydrates, lipids, proteins, nucleic acids, and adenosine triphosphate, or ATP, that you'll learn much about. Carbon has several properties that make it particularly useful to living organisms. It can form bonds with 1 to thousands of other carbon atoms and produce large molecules that have many different shapes. Due to this property, the body can build many different organic compounds, and each of those compounds can have a different structure and a different function. The large size of most carbon-containing molecules and the fact that most of them don't dissolve easily in water also makes them very useful in building body structures. Organic compounds are usually held together by covalent bonds. Small organic molecules can combine into very large molecules that are called macromolecules. Macro means large. Macromolecules are usually polymers. A polymer is a large molecule formed by the covalent bonding of many identical similar building blocks called monomers. Usually it's a dehydration synthesis reaction that causes this to happen. So let's think about those two words we just mentioned. Polymer, poly always means many and mono means one. So polymer, many units made up of monomers, one unit. So if you think of it like you're making a beaded necklace or a pearl necklace, the necklace itself would be a polymer and each individual pearl would be a monomer. Carbohydrates are the human body's primary and preferred fuel source. The monomer that makes up carbohydrates are monosaccharides. Mono means one, saccharide is sugar. So here you see some examples of different types of monosaccharides. If you join two monosaccharides together, you can create different types of sugar. For instance, if you put together a glucose and a glucose, you create the milk sugar maltose, or galactose and glucose, another milk sugar known as lactose. When you put many of those monosaccharides together, you can create a very large branched polysaccharide. Remember, poly means many. Most polysaccharides are going to be starches and cellulose. These do not taste sweet. They do not dissolve in water. The primary polysaccharide in the human body is glycogen, which is made up entirely of glucose monomers. Glycogen is a storage molecule where the liver will hook together individual little monosaccharides, individual little glucose molecules, and form a large branched polysaccharide known as glycogen. The storage molecule is very easily broken down in the liver when we need to increase our blood sugar, and broken back into simple sugars, little one sugars that can easily be put back into the bloodstream. So let's think about this. You eat a big meal, and that meal included several components that had carbohydrates and sugars, and not all of those were going to be used immediately. So your body decides to store some of those, and your liver takes little individual glucose monomers, one sugar, and hooks them all together into this long chain polysaccharide and stores it. Now, somewhere down the road, you're going to be without food for a little bit. It may be that you forgot to eat breakfast this morning, and you're hungry, and your blood sugar levels drop. Your pancreas can release a hormone that travels to the liver, and when it gets to the liver, it comes in here and breaks these bonds. When it's breaking these bonds, that's hydrolysis. Remember, hydrolysis, using water to break bonds. Hydrolysis reaction breaks all of these little bonds, and then we have all of these individual little glucose molecules that can be released into the blood, and blood sugar level raises. Lipids are fats that are found in the body. Excuse me. We have many different types of lipids, fatty acids, that are used to synthesize or to make other types of lipids in the body, and can also be used as a source to make energy, that ATP, the gas that runs our body. Triglycerides protect and insulate and also have an energy storage component to them. Phospholipids, which are lipids with a phosphate attached, are the major component of cell membranes. Steroids, this says blank, I don't know why. It should actually say that steroids are used to create cholesterol, as well as to make some of our vitamins, and used to build cortisol, the stress hormone, and estrogen and testosterone as well. When we look at fatty acids, we have long chain carbon molecules. Okay, so these molecules of carbon, or these atoms of carbon, are each completely saturated or full of all the hydrogens that they can hold. So this is what is known as a saturated fat, because the carbon backbone is a long straight chain. A mono unsaturated, or an unsaturated fat period, is one where the carbons are not completely full of hydrogens, but instead they form a bond between two of the carbons, form a double bond sharing those electrons. This allows that chain to bend a little bit. So this one has one double carbon bond, so it is a mono 1 unsaturated fat. These are the healthier fats as compared to the saturated fats. Okay, your saturated fats can all stack up on top of each other, sort of like building a block wall, whereas your unsaturated fats, because of that double carbon bond in there, they end up being sort of a bent or misshapen molecule, and those are not going to stack up and create fatty plaques in the blood. So just like we saw with the carbohydrates, we had a polymer, or the large molecule, that was made up of individual monosaccharides. So the polymer was carbohydrates, the monomer were the individual sugar molecules. With fatty acids, okay, or with lipids, we have a glycerol attached to a fatty acid molecule. So this would be a lipid. When you break this lipid down into its smallest pieces, you have a glycerol and a fatty acid. So the polymer is the lipid, the monomer are the fatty acids and the glycerol. So when we're digesting carbohydrates in our food, we break them down into monosaccharides so that we can use them to store energy and to create ATP. When we break down lipids or fats, we break them down into fatty acids and glycerol, and then we can use that to make the other fats that we saw in the body on the previous slide. So we can make triglycerides and phospholipids and cholesterol, and we can make some of the vitamins that are fat-based vitamins, okay? We also can store fat because it can be used to insulate the body, and also as an energy source, or as a source that we can use to make ATP when carbohydrate or sugar levels are low. Here you see an image of a phospholipid. Okay, phospholipids are the primary component of our cell membranes. The cell membranes surround all of our body cells and form a protective and selective covering, just like your skin does for your body. So phospholipids, you have the tails, you see the long chain hydrocarbon chains there, and a phosphate group that forms the head. So this shows you the chemical structure. This is more of just the little cartoony structure. And here you see how those phospholipids are arranged in a cell membrane. You can see that there are two layers of these phospholipids, and the heads are sticking out. So this would be outside the cell, this would be inside the cell. Now remember, our cells are filled with a fluid environment and surrounded outside by a fluid environment. So these phosphate groups that are a part of the head are polar. So that means they're hydrophilic, water-loving. The tails are the fatty part of these phospholipids. So tails are composed of the hydrocarbon chains that are hydrophobic, water-fearing. So you have these two sets of phospholipids with the polar heads that are near the water and the hydrophobic hydrophobic tails that are arranged away from the water, creating a phospholipid bilayer that makes up your cell membranes. Proteins give structure to the body, help to regulate processes, provide protection, assist in muscle contraction, help to transport substances throughout the body, and serve as enzymes that drive metabolic processes. There are many different types of proteins found in the human body. These are large molecules that contain carbon and hydrogen and oxygen and nitrogen. Some are also going to contain sulfur as a part as well. The monomer of proteins are amino acids. So remember how we used the pearl necklace analogy a while ago to describe carbohydrates? We can do the same thing with describing the structure of proteins. So proteins are big polymers made up of individual monomers, and the individual monomers are amino acids. So the amino acids, again, would be very much like the pearls in a pearl necklace, and the necklace itself would be the protein. So there are proteins that form structures within the body. You have lots of proteins that form fibers such as collagen and others that are found in connective tissues and also to help form structural framework in the body. You have regulatory proteins that work as hormones that drive physiological processes, help to control the rate and the way that the body grows and develops. Some of them act as neurotransmitters that mediate and cause responses in the nervous system. And some of them are contractile proteins as well. Contractile proteins are going to be the actin and the myosin that are involved in shortening muscle cells, which causes that muscle to contract and allows us to move. Other proteins serve an immunological function in that they can be formed into antibodies and interleukins that enhance the immune system response. They can carry vital substances when they are transport proteins, such as the protein part of hemoglobin that carries oxygen and very limited CO2 in the blood. And they can also be catalyst and have a catalytic function in that they drive and regulate biochemical reactions, such as serving as enzymes like salivary amylase that helps to start breaking down carbohydrates, or sucrase, an enzyme that breaks down sucrose, a sugar, and ATPase, which breaks down ATP. Now, as I mentioned, amino acids are the monomers that are going to form proteins. So the individual little amino acids all hook together, and when they do, they form peptide bonds. So the bonds that hold two amino acids together is a dipeptide. Okay, so a dehydration synthesis reaction removes water and creates a peptide bond. So now we have that glycine and that alanine hooked together, forming a dipeptide. Di means two, peptide is a small protein. Now, this would continue in making larger and larger and larger proteins depending on what protein was being made. You simply make another peptide bond and hook another amino acid to that. We'll look at protein synthesis in chapter 4, I believe, if not chapter 3. So when we look at the way that proteins are put together, there are 20 common amino acids that are used to create the primary structure of most proteins. The sequence in which those amino acids are hooked together, or if we go back to the pearl necklace, the way that we put them together on the chain makes the primary structure, which is simply what sequence you hook all the amino acids together. Now, you may begin with just 2 amino acids hooked together, as we saw on the previous slide, and create a dipeptide. Or there may be thousands of amino acids hooked together to form a very complex structure. Your genes determine what order we put those amino acids together, and we'll cover that shortly as well. Your secondary structure is created when these neighboring amino acids form hydrogen bonds. Remember we talked about hydrogen bonds being formed in one of our earlier lectures. So your secondary structure, all of these amino acids that we're hooking together have different characteristics. Some of them are polar, some are nonpolar. Some are charged and some are not charged. Some are hydrophilic, water-loving. Some are hydrophobic, water-fearing. So these different characteristics cause them to react together and form hydrogen bonds between neighboring amino acids. When that happens, the protein chain can then be twisted into what is known as an alpha helix, very much like an old-fashioned phone cord, or folded into a beta pleated sheet, very much like you would just fold a paper together back when you were in elementary school to create a little fan. Now, once we get those hydrogen bonds formed, then we have interactions that are going to cause further folding and twisting. So that chain that was twisted into alpha helices, and chains, that areas of the chains that form beta pleated sheets will continue to fold up into a functional protein. Okay, now this may be because you've got a negatively charged amino acid here on the chain and a positively charged amino acid here on the chain. What happens when you get a positive and a negative charge together? They attract. Remember, opposite charges attract. So this part of the chain would pull toward this part of the chain and create a folding in there. So that gives us our three-dimensional shape called the tertiary or third level shape of our polypeptide chain. And the final level of structure is what's known as the quaternary structure, where we have more than one polypeptide chain working together. So in this example, you can see a blue polypeptide chain and a gray polypeptide chain that have bonded themselves together into one functional protein. So it's only after we get to this tertiary or folded level, the three-dimensional shape of the polypeptide chain, do we have an actual functioning protein. All right, so let's talk a little bit more about these proteins now. An enzyme is a type of protein that serves as a catalyst in a reaction in a human cell. Okay, so enzymes, excuse me, enzymes cause a reaction to happen much faster than that reaction would happen without that enzyme driving that reaction. So enzymes are very highly specific. Hold on one second, let me check the next slide. Yeah, enzymes are very highly specific in that they have a particular binding site for a particular substrate. The substrate is what the enzyme is going to act on. Okay, so the enzymes have an active site that are going to bind to that substrate and cause an action to occur. Enzymes are very efficient. A lot of times they will catalyze reactions at rates that are from 100 million to 10 billion times more rapid than those of reactions that are very similar that occur without enzymes. So very, very efficient, and the enzymes are not going to be consumed in these reactions. They can simply unbind from that substrate and continue to control a reaction or continue to drive more and more and more reactions. Enzymes are subject to a variety of cellular controls in that their rate of synthesis and their concentration at any time can be under the control of a cell's genes, because enzymes are proteins. And remember, I mentioned that it's the genes that control what proteins are made. All right, let's move on. So here you can see an example of an enzyme. So the big orange thing here is an enzyme. Here's the binding site. So the enzyme is sucrase. The substrate is sucrose and water. So when those bind, they create a complex, and the enzyme breaks that disaccharide down into monosaccharides, glucose and fructose. Okay, so the enzyme catalyzed that reaction and transformed the substrate into the products. When the reaction is complete, the enzyme hasn't changed, and it is now free with its binding site open again, so that it can continue to catalyze that same reaction on more and more and more of the same substrate. All right, another category of macromolecules, organic macromolecules, are the nucleic acids. The one that you hear the most about typically is DNA, deoxyribonucleic acid. So DNA is going to store genetic information in the nucleus of body cells and helps to regulate most of the cell's activities. RNA, ribonucleic acid, helps to guide the formation of new proteins based on the information that's stored in the DNA. So when we look at a nucleic acid, RNA, DNA, and ATP, they're made up of individual nucleotides. So a nucleotide has a phosphate group, a sugar, and a nitrogen-containing base. Okay? So when you hook all three of those components together, it creates a nucleotide. Nucleotides are the monomers that make up the polymers DNA, RNA, and ATP. DNA has 4 nitrogen-containing bases: adenine, guanine, thymine, and cytosine. RNA has cytosine, adenine, guanine, and uracil substitutes for thymine. Okay? Now, we're going to get more into this when we look at creating new DNA and when we look at making proteins. Right now, just get the big picture of what a nucleotide is and that nucleotides are the building blocks of those nucleic acids. Okay? So we'll look just briefly at DNA. DNA is a double-stranded helix. So you have 2 curled molecules that wrap around each other. Each one of those has a phosphate group, a sugar, and a base, a phosphate group, a sugar, and a base, and it continues and continues and continues. So here we've got the sugar, the phosphate, and the base. The base, the sugar, and the phosphate. So going down one side of the double helix chain, and then the same thing on the other side of the double helix chain. You have your base and your phosphates and your sugars, your base, your phosphates, and your sugars. Hydrogen bonds are created between the 2 strands of DNA to hold them together. Now remember, we talked about hydrogen bonds being very, very weak bonds. So each strand consists of nucleotides bonded together by those hydrogen bonds. Each A on the strand binds to a T. Each T on the strand binds to an A. And C's bind to G's, G's bind to C's. Okay? So that's what's known as complementary base pairing. A's and T's always together, C's and G's always together. So if we do a quick comparison of DNA versus RNA, we look at what bases they have. DNA has adenine, cytosine, guanine, and thymine. RNA has adenine, cytosine, guanine, and uracil. The sugar in the nucleotide in DNA is deoxyribose. In RNA, it is ribose. Notice the similarity in those, ribose and ribose. Deoxyribose, D means to take off or remove, oxy, oxygen, so remove an oxygen from ribose. The number of strands, DNA is a double helix, RNA is a single strand. Your nitrogen complementary base pairing, A always goes with C, C always goes with G. In RNA, G still goes with C, but A, adenine, binds with uracil because remember, uracil replaced the thymine. We have to copy DNA and RNA. So how does it work? DNA replicates itself. RNA is made by using DNA as a blueprint. And then finally, the function. Your DNA encodes the information, holds the genetic information for making proteins, and your RNA carries that genetic code and helps to get that protein made. The final nucleotide or nucleic acid that we want to just briefly consider at this point is ATP. ATP is the principal energy storing molecule in the human body. It is adenosine triphosphate, so you have an adenine molecule with a sugar and 1, 2, 3 triphosphate groups. Okay, so there's a lot of energy held in these chemical bonds, and when we use this, we create ATP, I should say. We're storing energy in these chemical bonds that can later be freed and used to power metabolic processes in the body when we break those chemical bonds. And that takes care of chemistry for us.